When parents mate or are crossed, a Punnett square can be used to predict the phenotypic ratios in the offspring. A cross between a homozygous pure breeding dominant organism and a homozygous pure breeding recessive organism will result in an F generation of offspring with the dominant phenotype and heterozygous genotype. If these heterozygous offspring are crossed, the F2 generation will show the dominant and recessive phenotypes in the ratio of 3:1. The following diagram illustrates the genotypes of these cross and resultant phenotypic ratios. Mendel devised a method called a test cross to determine if an organism showing the dominant phenotype was homozygous BB or heterozygous B little b by crossing it with an organism showing the recessive phenotype BB, lowercase. If any of the offspring showed the recessive trait genotype lowercase b, the organism in question had to be heterozygous. Some genes have more than two possible forms, multiple alleles. For example, there are 3 different alleles that determine blood type in humans. The alleles and LB show codominance, phenotype AB. The allele I is recessive to both capital IA and capital IB. These alleles form the following phenotypes and genotypes. Another form of allele interaction is incomplete dominance. If both alleles are present in the genotype and neither is dominant, then the phenotype will be a mixture of the two. For example, if a pure breeding red flower and a pure breeding white flower are crossed and the F1 offspring are all pink, then incomplete dominance exists. A cross of 2 of the Fs would produce an F2 generation with a ratio of 1 red, 2 capital R, to 2 pink, capital R, capital W, to 1 white, capital W, capital W. Mendel also completed crosses involving 2 different traits, dihybrid crosses, and found that they also produced characteristic ratios. P1 parents, pure breeding, for 2 traits were crossed, producing an F1 generation of all dihybrids. When 2 of the F1 dihybrid offspring were crossed as the P2, the F2 offspring occurred in the phenotypic ratio of 9:3:3:1. Mendel explained these results by stating that the gene for each trait was inherited independently of the other gene, law of independent assortment. The alleles for each gene assort independently as long as they are located on different chromosomes. Some traits are polygenic, meaning that 2 or more genes determine a single trait. Examples are skin color and height. Polygenic traits produce a wider variability in phenotypes than single gene traits. Some genes are epistatic, meaning that 2 genes interact to form different phenotypes. The environment also plays a role in whether or not genes are expressed. Most genes are affected by the environment that exists inside or outside the body, such that a phenotype may or may not be produced, even though the corresponding genotype exists. Inherited conditions can be mapped through the generations using a diagram called a pedigree. The pedigree indicates the generation of an individual, whether they are male or female, the position of the individual within the family, the mate of the individual, and whether or not they are affected by the condition. Both dissolved annulled. Sex linkage. Most genes are found on the 22 pairs of autosomes, non-sex chromosomes. The 23rd pair are sex chromosomes that determine an individual's gender. Females have 2 X chromosomes. Males have an X and a Y chromosome. X and Y chromosomes are not homologous. Genes that are found on the X chromosome are not found on the Y. It exists in an app, which is rejected because of fraud. Phenotypes associated with genes on autosomes are found equally in males and females. Genes that are found on sex chromosomes, sex-linked genes, produce phenotypes that are found in different ratios in males and females. X-linked genes are found only on X chromosomes. The Y chromosome does not contribute to X-linked trait. X-linked recessive traits such as hemophilia and colorblindness are more likely to show up in males than females because males do not have a second X chromosome that could counteract a recessive allele on an X chromosome. A cross of a male without hemophilia, XHY, and a woman who is a carrier for hemophilia, XH, would not produce any hemophiliac daughters. Conversely, because females have two X chromosomes, X-linked dominant traits are more likely to show up in females. A cross between a male with hypotrichosis, XCHY, and a normal female, XCH, would not produce any sons with hypertrichosis. A father will always pass his X chromosome to his daughters. His Y chromosome is always passed on to his sons. A mother will always pass on X chromosome to neither sex of her offspring. A carrier is an individual who has an allele that does not appear in the phenotype. Because females have two X chromosomes, females can be hidden carriers of an X-linked recessive allele. When carrying out a cross involving a sex-linked gene, both the sex chromosomes and the alleles on the sex chromosomes must be indicated. Determining the type of inheritance operating can be difficult, but there are indicators of each type. A condition is autosomal if there is no significant difference between the genders in the frequency of a phenotype. A condition is sex-linked if there is a significant difference in the incidence of the condition in males and females. A condition is autosomal recessive if unaffected parents are producing an unaffected child. Gene linkage and chromosome mapping. Because there are 20,000 human genes on only 23 chromosome pairs, many genes are linked together on each chromosome. When two genes are linked on the same chromosome, independent assortment cannot occur because the linked alleles are not free to separate from each other.